Welcome back, everyone, to our bi-weekly speaker series. This virtual seminar series is designed to reconnect researchers and bring expert content directly to your sofa. Today's talk will be presented by Dr. Josef Penninger. Josef is the Director of the Life Sciences Institute and a Professor in the Department of Medical Genetics at the University of British Columbia, where he currently holds the Canada 150 Research Chair in Functional Genetics. Josef also holds an appointment as a Professor of Genetics at the University of Vienna, where he retains an active research program, and he also is joining us today from Austria. Josef's accomplishments include pioneering insights into the molecular basis of osteoporosis, breast cancer, and metastasis, and he has published over 60 publications in Cell, Nature, and Science. Today, Josef will be covering his recent Cell publication with a talk entitled Soluble ACE2 as a Rational Therapy for COVID-19. Josef, please take it away.
Yeah, so thanks so much for having me. So I try to share my screen now. I guess that's what works. So it's great to meet you all online. Apologies that I cannot be with you in person in Canada, but I'm actually let me see. Here we go. I actually got stuck in Vienna. I wanted to go back, but just one day before I wanted to go back, the borders came down. And so I was not allowed as a non-Canadian citizen back into Canada. Today I want to tell you about how we discovered ACE2, how this all relates to COVID-19, and basically the basics, how we believe this all can fit together. That's my new job in Vancouver. You got already the introduction, so I keep it brief. I'm now running the Life Science Institute at UBC, but I have also some people left in Vienna. So early on, when I was a young PI in Toronto, we were interested in the development of fly heart, and there was this particular marker called EVE, and we screened P-element fly lines for genes which would regulate heart development in the fly larvae. And then we realized that flies had actually two orthologs of a gene called ACE, Angiotensin converting enzyme. And because flies having two orthologs, then Mike Krakauer, the Toronto kid who now works for biotech in Boston, thought if flies have two genes, then mammals should have two also. So Mike cloned the second ACE gene, which we called ACE2. If you can see, this was from our first paper. It's expressed in the kidney, in the lung, in the heart, in other tissues. I should also mention we did not publish the sequence first, so our two other groups did, because we had worked on the gene, and the reason why we did not publish was simple, because we wanted to make a knockout mouse and see the in vivo function. And this was the phenotype. This is a normal echocardiogram of a mouse. And if we knock out ACE2, then you see this mice develop spontaneous, at least in this mouse background, spontaneous impairment of heart function. And what was really interesting, when we knocked out ACE2, you see in the middle there's spontaneous impairment of heart function, on the top it's normal. You can also see, which is quite interesting, it's actually on the X chromosome, which could have an influence on gender differences and hypertension, and of course also for COVID-19, which I will get into it. The ACE2 knockouts have this impaired heart function, and if we knock out ACE on this background, then we see total rescue of the phenotype. So after many years and many papers we did, and of course other researchers did, we got together this pathway. Angiotensin 1, a decapeptide, gets cleaved by ACE, and this was known before we had discovered ACE2, which makes an octapeptide called angiotensin 2, which acts on two G-protein coupled receptors that regulate blood pressure, heart function, kidney disease, fibrosis of multiple tissues. So there's the renin angiotensin system, of course, we knew for a long time, and it has a major physiological role in our body. So what we discovered and published first in vivo and showed the first in vivo function as a negative regulator of the system was ACE2. And how it works, ACE2 is actually cleaving the last amino acid of Ang2 off, and by doing so, inactivates the system. ACE2 is also an unspecific protease, so it actually cleaves apelin, bradykinin, neurotensin, dynorphin, so it's not totally specific for the renin-angiotensin system. And by doing so, ACE2 is the good guy in the system. If we knock out ACE2, the mice get liver fibrosis, they get more kidney disease, and I showed you the data on the heart. So this was fine, so identifying really for the first time in vivo a negative regulator of the system. And then we realized ACE2 was strongly expressed in the lung, which at this time, this was 2002, around 2002, didn't make much sense. So the Western blot and wild-type mice and ACE2 knockout mice, so ACE2 was gone. So we were wondering what's the function of ACE2 in the lung, and we looked for lung function, there was no difference. And then we started to collaborate with Art Slutsky in Toronto and a postdoc of Art's, Yumiko Imai, who actually then went with me, this was around 2002, with me to Vienna to work with me in Vienna. And Yumiko, over many years, had developed a very good model of acute lung injury in mice. At this time, very few people had been actually working on this field because it was very difficult to actually set up systems like this. So the question was, would ACE2 play a role in acute lung injury? Because we had this model. And the model is acid aspiration, but with other models of lung injury. So on the top, you already see an injured lung after acid respiration, 3-4 hours after the acute injury. There's already bleeding in the, let's say, wild type mice. When we put in Evans blue into the belly, so we look for leaking blood vessels, you see already a little blue. However, if we do this in ACE2 knockout mice, the mice get super severe lung injury, inflammation, bleeding all over the place, and the entire lung gets blue, so the blood vessels just start to leak like crazy. And then we genetically map the whole phenotype. So here, wild-type mice, this is already injured lungs. ACE2 knockout mice get super severe disease. If we knock out both alleles of ACE, so one allele of ACE, on the ACE2 knockout background, we see rescue of the phenotype. So we came up with this pathway that lung injury, various types of lung injury, turn on the renin angiotensin pathway. Ang2 is shooting up, and this has now been shown in many, many cases of lung injury, including of COVID-19 now. Ang2 goes up, and via the AT1 receptor triggers edema, inflammation, a leaky blood vessel, leading to very severe lung edema, severe inflammation, severe acute respiratory distress syndrome, ARDS. So this is where we were, and then all of a sudden, Mike Fasan from Boston identified, and this was around the time when the SARS virus was striking Toronto, and I was still there, I was just moving to Vienna, and of course the first SARS epidemics from China going into Canada. This time at the end infecting 8,000 people, 400 in Canada, 40 of them died. And Mike identified ACE2 as a candidate receptor for the spike protein of the SARS coronavirus. The spike would bind to ACE2, leads to fusion and entry into the cell. So, but there were many other candidate receptors identified, for instance, L-signs, or the slur of papers out there of candidate receptors for the SARS coronavirus. So the question was, is ACE2 really critical? Could there be other receptors, which of course could be possible, considering viruses sometimes in multiple receptors? Since we had the first knockout mouse made, we immediately sent our mice to China, to Peking Uni Medical College. They infected the mice with the SARS coronavirus, an intranasal infection. And as you can see in wild-type mice, the virus grows. In ACE2 knockout mice, there's no growth of the virus anymore. And here is pathology. The SARS virus makes some mild pathology, nothing like in humans, but there's some pathology. ACE2 knockout mice are totally healthy. But this experiment was very important because it shows in vivo that ACE2 is the essential receptor for the SARS coronavirus. No ACE2, no infection. The other thing we saw, if we infect mice with the SARS coronavirus, if you look in the lung and the heart and Western blots for ACE2, in non-infected mice, you see, of course, ACE2 band here. However, if there's an infection, and in the knockout mouse, it's gone, obviously, but there's an infection with the SARS coronavirus, ACE2 disappears. Basically, the SARS infection leads to a mouse which doesn't express ACE2 anymore, like an ACE2 knockout mouse. So the question then was, could we explain via ACE2 why SARS became a killer? Would ACE2 be regulating lung failure explain the lung disease in the SARS infection, of course, now COVID-19? This was the experiment we did. We have this lung injury model with ACID. So this is already change in elastance, change in lung function. On the bottom is normal lung, and the middle is ACID injury, severe injury. And then we gave the mice Spike. So we cloned Spike from the SARS virus, put it in, and the mice got much worse. So to put this all together, what we basically had found over some years, of course, with help of many collaborators, that lung injury, pneumonia, acid aspiration, sepsis, SARS virus infection, and now COVID-19. The virus causing COVID-19 turns on the renin angiotensin system. ACE generates Ang2. Ang2 via the AT1 receptor drives, and probably also via bradykinin receptors, drives leaky blood vessels, severe lung injury, inflammation, you name it, edema, the blood vessels open. You're probably aware that, for instance, ACE inhibitors lead to dry cough and lung edema. The reason is actually here in the system turning on bradykinin receptors, so they're also involved. So it's not the only system. And ACE2 is the negative regulator. Of course, if you have ACE2, you cleave a lot of Ang2, you reduce the injury, you kind of make it milder. No ACE2, severe injury. Now the virus came along, hit ACE2 to infect cells. By doing so, it actually takes ACE2 into the cell. By endocytosis, ACE2 disappears, the virus gets in, but you now not only have the virus infection and the entry into the cell, but also now you lose protection. Ang2 becomes prominent, and one loses protection. And this is what we believe is actually the reason why the SARS virus became a killer. So the question then was, could we use this for a new therapy? Because if ACE2 is so critical, let's clone a recombinant human ACE2, a soluble form, put it into as a therapeutic option in our mouse model of acid injury. Here you see this, acid control, and then we do acid injury. If the red arrow here, recombinant ACE2, and we can stabilize the disease. So based on all of this, that ACE2 is the critical in vivo SARS receptor and protects from lung failure, we actually developed via biotech, and I declare myself a conflict of interest right here, a recombinant human soluble angiotensin converting enzyme number 2 to basically use it to protect lungs from lung injury. Of course, at this time, nobody really cared so much because SARS had disappeared. And this was our data. This molecule actually went already into 89 humans, healthy volunteers, and also people in phase 2 trials, people with pulmonary hypertension and lung injury. This was a pig model, LPS-induced pig ARDS, published in Critical Care Medicine. The oxygenation of the blood goes down, the PaO2, and it goes down, and you see in the IV bolus injection to actually stabilize the disease. So the reason why we developed this, as I said, and then actually we know the PK, we know the ACE2 is stable, so we did actually 10 years, more than 10 years of drug development. The reason why we did this is because we wanted to develop a molecule which helps against acute respiratory distress syndrome, which is more or less the syndrome in response to many diseases, many pathogenic triggers like sepsis, gastric acid aspiration, the first SARS coronavirus, Spanish flu, avian flu. By the way, we co-authored the paper where avian flu triggers lung injury, turns on Ang2, and actually giving ACE2 can alleviate the lung disease, anthrax, smallpox, bioterrorism, you name it. So there are many ways which lead to this with this syndrome, which we call acute respiratory distress syndrome. So this was interesting. And then, of course, for many years, people didn't really care much about ACE2. The heydays of ACE2 were in the early 2000s. And then, of course, the SARS coronavirus #2 came along. And when the virus was sequenced, it became very, very soon clear that ACE2 should be the critical receptor, because the virus spike protein has 88% identity to the spike protein of the first SARS virus. There was another coronavirus which became very lethal called MERS, and this has around 56% identity. MERS uses another receptor called DPP4. So, so it was very clear that ACE2 should be the receptor. So this was actually one of the papers, and the many, many other papers which show this. These are human, the HeLa cells, which don't have ACE2. So on the bottom here, because they don't have ACE2 in red, the virus cannot infect. If one puts in human ACE2, this is human ACE2 in green expressed, the virus can get in. Bovine ACE2 works, swine ACE2 works, civet ACE2 works. Mouse ACE2 does do nothing. So if you overexpress mouse ACE2, it doesn't bind. There's some amino acid changes. And actually this new coronavirus 2 has higher binding affinity, especially in the not in the on, but in the off KD, which allows higher affinities and higher binding to ACE2 as compared to the first SARS virus. I should also say there are multiple other viruses out there, coronaviruses, in the group 2B coronaviruses. Many of them live in bats, but also in other animals, which could, based on the sequence, use ACE2 to infect us. So these are not the only one which can do this. So there are many out there, but they have not jumped yet from animals to humans. This was the structures very fast. So ACE2 went from nearly being ignored to probably being one of the most researched molecules on the planet. So from artificial intelligence to companies to many, many researchers in essentially every university working on this now. Within few weeks and days, there were crystal structures and cryostructures. So this was one of them. On the bottom is the virus spike protein, 3 molecules of spike. In green is ACE2. And this is the cryostructures here on the top. This little dot, this on the eye is the ACE2 binding to the spike. And this is just some again, to show this. So it's absolutely clear ACE2 is the critical receptor for the SARS coronavirus 2, and it's essential for entry of the virus into the cell, which does not exclude there might be other co-receptors out there. I'm pretty sure there are, but clearly ACE2 is the critical one. So the first question we asked now in a paper we published 2 weeks ago online, and it will come out now soon, is since we had this soluble ACE2 molecule, which is actually now moving into clinical trials, can this actually reduce the SARS infection? The reason is this: if the SARS coronavirus 1 or 2 come along, it binds to ACE2. This leads to internalization of ACE2 together with the virus, membrane fusion, the viral RNA spills out, makes its pathology, leading to the disease we call SARS or COVID-19. So this was the idea. We saw a soluble ACE2 molecule, fully human recombinant. We cleaved off this transmembrane domain. And we know it's fully active. It's glycosylated, made in CHO cells. So it basically, with the binding affinity is like an antibody. It's 1.6 nanomolar. So it's very, very high affinity to the spike protein. So we basically would assume that's like a neutralizing antibody, super specific, because this is how, of course, the virus enters cells, and this should soak away the virus, less internalization, improve disease. So it would actually be an antiviral blocking virus entry. So we hooked up with a group in Sweden, Ali Mirasimi. I've worked with him for a long time. We worked with him on Ebola, Zika, Marburg virus infections. Ali isolated, as you can see, on the 7th of February 2020, the first SARS coronavirus isolated in Sweden. In Jönköping, this is how it looks in EM. So this is our virus, and it's the clade. And this is the experiment we did. So we actually infected viro cells with different plaque-forming units here on the top, from 10 to the 3 to 10 to the 6, and then gave them different concentrations of the ACE2 molecule, which went to clinical testing and is now going into COVID-19 patients. And as you can see, it has very strong effects suppressing the virus infection. It's the dose response. Of course, if you infect with more virus, you need more ACE2 to get the effects. And on the bottom, we did the same experiment with mouse ACE2, and it doesn't do anything because it cannot bind to the spike of the virus. So this is a live virus infection, and the effects of ACE2 is the reduces viral load by a factor of around 1,000 to 5,000 times. We compared this now, and which is not published now, with remdesivir. So remdesivir has some good effects, but at very high doses. At low dose, it does basically nothing. And at this high doses in our assay is very toxic for the liver. So remdesivir does something in the same assay at very high dose, and we believe it's maybe not safe because of liver toxicity. But of course, we will need to see this. And then there's the other issue. We all know, and you probably all read this recent article in Science, but we knew anyway, it's just well put together that COVID-19 might start in the lung, but it's a multi-organ disease affecting multiple tissues from the kidney to the heart to the intestine. And of course, ACE2 can possibly explain this. Actually, it does explain it because ACE2 is expressed on the heart, on the blood vessel, on the endothelium, in the central nervous system. ACE2 changes with age. There's now many papers coming out. Gender, it maps to the X chromosome. It changes with cardiovascular disease, with diabetes, with smoking. This was from one of our papers where we showed ACE2 being expressed in the luminal surface of the intestine. Now the data is coming out. Many COVID-19 patients have diarrhea. The virus replicates here because it makes sense. It's lots of ACE, and ACE2 is heavily expressed in the proximal tubules in the kidney here in green. So they've, of course, begs the second question: can ACE2, via its enzymatic function in the RAS and probably in the bradykinin system, protect from organ damage? Because this is the negative regulator of a system which triggers organ damage in multiple systems in thousands of papers. So this is one of our papers where we actually show this in heart failure. Wild-type mice, they actually get this heart failure protocol. And by aortic bending, in light blue, they get worse disease. And if we give the recombinant ACE2 protein, the same which actually goes now in patients, has been tested in patients, you can see it alleviates the symptoms. The same is for liver fibrosis, severe liver fibrosis. This is not our work, we just provided the mice. Christoph Österreicher showed this. Recombinant ACE2 reduces the symptoms as expected, blocking Ang2. This is a model of diabetic nephropathy in Akita mice, insulin 2 mutants. This is albumin excretion in the urine. This is collagen deposit in the kidney. And as you can see, recombinant ACE2, which we call APN01 as a drug candidate, reduces the disease. So in basically all the models we tested, as expected from the biology and physiology, actually the recombinant ACE2 protein reduces disease. And this was also what we then tested in our paper, which was published 2 weeks ago. We actually engineered human tissues because we wanted to have human relevant tissues and infect them, because everybody just infected viro cells by the cell types. So we engineered human blood vessel organoids, which were published last year in Nature. And in B here, you can see we infect them with different doses of the virus. The virus indeed can grow. This is testing for viral RNA. 3 days post-infection, 6 days post-infection. You can see the more virus you give, of course, the more virus grows. In C, we actually get progeny virus out, meaning if we infect these blood vessels, then the virus actually assembles and we can take it to infect other cells. And in D, when we give recombinant ACE2, we can significantly reduce the viral load in this organoids. You might have seen 2 days ago, there was a paper published, and this was really interesting in the Lancet by pathologists from from Switzerland, from Zurich, in 3 patients who unfortunately died of COVID-19, and there was an autopsy. And in all of them, and they claim there's a systemic infection of the endothelium leading to systemic vasculitis, which could, of course, explain why some of these patients deteriorate so fast, because if there's a blood vessel infection, and they directly show the virus is in the blood vessels by electron microscopy. So this follow-up paper where we show we can directly infect them, and so we know now it's truly happening in patients. And COVID-19 at late stages, as they claim, might be actually a vascular disease. The second experiment we did was together with Núria Montserrat in Barcelona. She made from stem cells and iPS cells very unique kidney organoids with tubular structures, which are here with marker in green LTL for proximal tubules. This is exactly the place where ACE2 is expressed, which also can show. So there's ACE2 expression also in the blood vessel organoids. Actually, we did this together with Stem Cell Technologies in Vancouver to help us. This is Núria's work really in Barcelona. Again, in B, we infected them with the virus. The virus can grow, and we have viral progeny in C. This on the bottom here was not in our paper. This was our first experiment in intestinal organoids. There's lots of ACE2, and as you can see in E, we're still working on this. The virus grows like crazy, as you would expect being ACE2 around. So this is where we are. So basic from discovering, making the first ACE2 knockout mouse, linking it to lung failure, explaining why the SARS virus, and of course now the SARS virus 2 became killers, hitting ACE2. Because ACE2 has multiple function, multiple protective function in multiple systems. So this explains tissue distribution, as they also write in the recent Science paper. And of course, probably might be directly involved in tissue damage. Of course, there's many other aspects of tissue damage, cell death, inflammation, you name it, hijacking of metabolic pathways. So where we are now in clinical development is this. APN01 is soluble recombinant ACE2. All the data I showed you, new data and the data in mouse models to alleviate disease, is exactly with this molecule. And this molecule has been tested in 89 patients, healthy volunteers and patients in phase 2 with lung disease. And 2 weeks ago, we got approval by the European authorities in Germany, in Austria, and in Denmark. And we're actually talking to many more now in the UK, for instance, in Belgium, in Stockholm, in Spain, to go for a placebo-controlled, double-blinded phase 2 clinical trial. In China, we're still talking to the authorities. The reason why we do this is, first, it should reduce the viral load, as I showed you, and secondly, it actually should protect from organ damage. Of course, there will be no magic bullet. ARDS, we have worked with ARDS. It's a nasty disease, many aspects of the disease, but it's actually, you know, the logical pathologic pathway. It's the entry of the virus to infect us, and it protects many tissues because of its basic physiology. Also, what's important, so this will be done in 200 patients. 100 will get placebo, because placebo meaning standard of care, and 100 will get the drug. In severe COVID-19 patients, you might know the stages. Stage 1 is mild, stage 2A is moderate, stage 2B is already in severe involvement of the lung. People get hospitalized. And stage 3 is then the deteriorate on the ICU. So we will treat stage 2B patients. The other important aspect is here on the bottom. Actually, the molecule we have is, it has still both functions, because the virus would bind here, but the catalytic activity, the enzyme function, protecting tissue, is actually on the other side. So the virus binding, as far as we know, does not change. Catalytic functions and catalytic changes, like when we make a point mutant to kill this, does not change how the virus binds. So we basically should get the double whammy. And of course, now we're setting up to carefully determine if this works or doesn't work. I'm quite amazed about all these claims of some drugs which work, but they've never been properly tested. So what's really needed now is to have careful testing of drug candidates, because at the end of the day, we need to provide some medicines which actually work and are not anecdotal. With this, I leave you Mike Krakauer, Toronto kid, cloned ACE2 in my lab. Again, we published the first knockout mouse. We had it in the lab for a long time. The sequences were published by others first, so the credit to them. Yumiko Ima, KJ Cooper linked the SARS infection to lung failure, were really the first ones to actually ever show this, together with Art Slatsky in Toronto. Rainer and Alex developed blood vessel organoids. Nuria Montserrat in Barcelona helps us and does amazing work with kidney organoids. And we do all the infectious work in the super high security lab of Ali Mirasimi. Vanessa, a postdoc in his lab, is actually doing the work. She's the one who puts her finger to these viruses. This is downtown Stockholm. This is it's like the Winnipeg for lab for dangerous viruses for Northern Europe. So Ali is one of the guys. If the Ebola breaks out, he's on the ground in Africa. So it's very good. It's a very dangerous virus. So we have it in safe environment. And of course, many more people helped us. Thanks for listening. And I guess with this, I stop.
Okay, thank you so much, Josef. So maybe some questions. Are you good for answering? Some questions right now. So the first question we have is from an anonymous attendee, and it is taking ACE inhibitors not recommended if one is infected with COVID-19?
Oh, geez, that's a loaded question. This is a raging question in the literature. I think one of the reasons came actually from our first paper, where we showed that the AT1 receptor drives this lung injury. Now there's the ACE inhibitors, which of course block generation of Ang2 and everything downstream, and the ARBs, so angiotensin receptor inhibitors, which of course act downstream of Ang2. So we know peptide maps, so I can answer this with science. If you actually do the science, and we have developed very fine mass specs in the serum of people. If you put an ACE inhibitor, as you expect, everything, all the peptide maps downstream are blocked, obviously. If you put now ACE2, then ACE2, what it does, it diverts Ang2 into angiotensin 1 to 7 and Ang2 5, which is a particular receptor called MAS, which has positive effects. If you now put an ACE inhibitor and ACE2, ACE2 doesn't have any substrate anymore. So that's why it doesn't work here. So one of our exclusion criteria for our clinical trial was the people who have on ACE inhibitors are not allowed being part of the trial, because of course ACE2 wouldn't have a substrate. So I think the question is still out there, difficult to answer. We need some careful clinical studies to actually answer this. It's also a loaded question because there are many people who have heart disease and hypertension or inhibitors. Now to take them off and risk strokes and heart attacks is a risky business. But I know there's in Vancouver, for instance, a group which will do a clinical study to addressing this exactly.
Okay, thank you. So the next one is from Rens de Groot. Could you elaborate a bit, please, on asymptomatic patients and people with very mild symptoms? Is it as simple as them having very low ACE2 expression?
It's very hard to tell. Maybe not. We don't know. Of course, it starts. We get infected. If I would be infected and close to you, you might get exposed to my droplets. It starts in the nose and throat, and this is where it might end for many people, which might, a little cough or being asymptomatic, and of course, it sometimes goes deep into the lung. So the deep lung pneumonia we can explain with the expression of ACE2, because it's very deep in the alveoli, type 2 alveolar cell. ACE2 is also in the nose and some of on our throat. So, but if this really explains this, I don't know. The same thing for age and gender, for instance, we don't know.
Okay, thank you. So the next question is also from an anonymous attendee. Do any small drugs that bind ACE2 block viral binding? Can these be developed?
Oh, geez. We did this drug development for 10 years now. So we are basically 10 years ahead. But I get every day's email from people and companies who do exactly this. They want to make small molecules, to make, I don't know, cyclic peptides. There are AI companies, basically every AI company on the planet is predicting better ACE2 molecules and molecules which would interfere with this. Companies who make vaccines actually targeting exactly this interaction site. So yes, there are probably thousands of people doing exactly this right now.
It's proved to be a difficult target to drug then?
I don't know if it will prove to be difficult, but that's what they're doing. Nobody did target it because, of course, SARS disappeared and nobody cared anymore.
Okay. So the next one then, another anonymous attendee. How many milligrams of ACE2 protein is required to treat one patient?
In our clinical study, based on pharmacodynamics in phase 1 and phase 2 trials, we will use 0.4 milligram twice a day in intravenous infusions, and the treatment will be for 7 days. So that's basically it. We're actually working now and might want to do a phase 1B pilot trial in few patients, probably in Barcelona and in Stockholm, to reduce the dose and have longer term infusion to basically still get the effect, but don't get just the bolus in the hammer, but distributed over time. But what we will use in our phase 2 clinical trial is 0.4 milligrams every 12 hours. I should say, no, 0.4 milligrams per kilogram of the person.
Right.
That's the exact dose per kilogram.
Okay. What is the plasma half-life of APNO1?
The plasma half-life in humans is around 11 hours. In rhesus monkeys, 10.5 hours. In rats, around 9 hours.
Okay. And the final question.
So that's why we're actually dosing it every 12 hours.
Sorry, the final question we have is, which part of the liver may be affected by SARS-CoV-2? I presume they're referring to...
The liver is hard to tell. It might be just the vasculature, which and then leads to severe inflammation. The hepatocytes, as far as we know, don't really express ACE2, so let's see how this plays out. So the liver damage might be indirect, but of course we cannot exclude it. That there might be something else, or maybe there's a second receptor somewhere.
Okay, there's one final question come in. Is anyone looking at developing a drug that targets the spike protein to block its binding site for ACE2?
Oh yeah, that's basically most of the vaccine guys, who do single antibody vaccines. You might have heard GlaxoSmithKline invested in a California company $300 million to do exactly this. So, and I'm sure there are many more out there. Because what if you target something in the virus with an antibody, what would you target? Probably the interaction site to its receptor.
Okay. Do you have time for one more, Joseph?
No, please go ahead.
Is there any data about renin volumes, renin being at the top of the chain, and COVID-19 disease progression?
That's a really interesting question. There's actually data from China which show that Ang2, the higher the Ang2 levels, the worse the disease. There are also data on avian flu, the higher Ang2 levels, the worse the disease. Of course, renin would drive this. Of course, in kidney disease, there's lots of renin, and so we have the high levels. We actually did 2 patients on compassionate use, so we never published this, we never talked about this, about 2 patients with COVID-19. So one had unbelievable high levels of Ang2, very sick patient, and the guess is actually there was some already kidney damage, and because the renin kicked in, and that's why it was driving it up. But we have not systematically measured it. But this, of course, will be all secondary endpoints in our clinical trial. Measure all those peptides, and so we have a very careful setup. The measurement of the renin angiotensin system from renin to ACE levels to ACE2 levels to basically all the 6-7 peptides in the pathway. It's not just Ang2. There are many other peptides in the RAS pathway which are relevant. Ang1 to 7, Ang1 to 5. We will measure all of them to have it set up, and that's very important because we're also measuring the ratios, because Ang2, and you naturally you might have a sort of different level of Ang2 like me, so we have to compensate for all of this.
Okay.
But that's taken care of.
All right, so we've still got some questions coming in. I'll just do 2 more, and then we'll finish. Could you comment on the large difference in mortality rates between SARS and COVID-19, given the similarities in genome sequence and the viral entry via ACE2?
Hard to tell. In SARS, as far as I know, so when it started, also in Canada, the lethality rates of SARS were 2%. So when everything was over, and there were many people still in intensive care, it actually jumped to 10%. If one looks at the Johns Hopkins map, and that's probably very biased in reporting and so on, I think the lethality at the moment from confirmed cases to actually deaths is 6.5%, which is very surprising. But of course, there are data now coming out, for instance, in Germany, that many people have the virus are asymptomatic. So I think the rates and the morbidities and mortalities, we can really only tell when the dust settles and we really have all the data in.
Okay.
So it could turn out to be like the flu with 0.1% because we're all infected and most of us didn't realize it. Or it actually could be around 4 or 5% because many people are still in the ICU, won't recover. I talked to the ICU doctors, and they tell me they've never seen anything like this. So it's not like ARDS, the typical ARDS. Also, intubation and giving a lot of oxygen is probably very toxic for them. So they're still really struggling with the disease and how to keep it. So let's see.
Okay. Do you think the use of soluble ACE2 could lead to development of autoimmune response towards cells expressing higher levels of this receptor?
I don't believe so, because ACE2 is expressed in many tissues on the endothelium. The immune system would be exposed to it and would see it all the time. So what we're basically doing, we did not engineer an FC fusion or a particular linker. It's the natural molecule which is just cleaved off at the extracellular domain, where it's also naturally cleaved off by ADAM17. There's actually soluble ACE2 circulating in our serum, not much. So it's hard to imagine this would happen. Of course, if you put an FC fusion or dimerize it in certain ways, it could happen, or change it from the natural ACE2. It could happen, but we have basically natural ACE2, which also floats around in our body.
Okay.
And it actually is a dimer. It's a natural dimer. It naturally dimerizes.
Okay, well, thank you. And thank you for answering all of those questions as well. And on behalf of the audience, I'd like to thank you for a superb talk and a hugely educational Q&A as well. Thank you. So,
thank you.
I would just like to give you a heads up before everyone leaves that our next Stem Cells from the SOFA event will be held on 1 p.m. EST on May 6th. And it will feature Dr. Christina Nostro. She's the Harry Rosen Chair in Diabetes and Regenerative Medicine Research at the McEwen Stem Cell Institute at the University Health Network in Toronto. Christina's talk will focus on modeling human pancreatic development with embryonic stem cells. Okay, until next time, thank you very much.